Media Advisory. Tuesday, May 9, 2023. Findings may aid design of targeted drug delivery into the brain and central nervous system. Researchers at the National Institutes of Health and colleagues have developed a zebrafish model that provides new insight into how the brain acquires essential omega-3 fatty acids, including docosahexaenoic acid D, H, and linoleic acid at L. Their findings, published in Nature Communications, have the potential to improve understanding of lipid transport across the blood-brain barrier and of disruptions in this process that can lead to birth defects or neurological conditions. The model may also enable researchers to design drug molecules that are capable of directly reaching the brain. Omega-3 fatty acids are considered essential because the body cannot make them and must obtain them through foods such as fish, nuts, and seeds. D, H, levels are especially high in the brain and important for a healthy nervous system. Infants obtain D, H from breast milk or formula, and deficiencies of this fatty acid have been linked to problems with learning and memory. To get to the brain, omega-3 fatty acids must pass through the blood-brain barrier via the lipid transporter MFSD2, which is essential for normal brain development. Despite its importance, scientists did not know precisely how MFSD2 transports D, H, and other omega-3 fatty acids. In the study, the research team provides images of the structure of zebrafish MFSD2, which is similar to its human counterpart. The snapshots are the first to detail precisely how fatty acids move across the cell membrane. The studied team also identified 3 compartments in MFSD2 that suggest distinct steps required to move and flip fatty acids through the transporter as opposed to movement through a linear tunnel or along the surface of the protein complex. The findings provide key information on how MFSD2 transports omega-3 fatty acids into the brain and may enable researchers to optimize drug delivery via this route. The study also provides foundational knowledge on how other members of this transporter family, called the major facilitator superfamily MFS, regulate important cellular functions. The study was led by Doreen Matthews, Ph.D. of National Institute of Health's Eunice Kennedy Shriver National Institute of Child Health and Human Development NICHD, and Tamir Ghanem, Ph.D. of the University of California, Los Angeles. Additional funding for the study was provided by National Institute of Health's National Institute of General Medical Sciences NIGM and the Howard Hughes Medical Institute. Doreen Matthews, Ph.D., Head, and Louis Tung Fat Lai, Ph.D., Postdoctoral Fellow, Unit on Structural Biology, are available for interviews. To arrange an interview with Dr. Matthews or Dr. Lai, please email mishpress@mail.gov or call 301-496-5133. Nguyen C. Lay H. Tiv. Fly LTF. L. Lipid flipping in the omega-3 fatty acid transporter. Nature Communications D. OI 10.1038/41702-72023. About the Eunice Kennedy Shriver National Institute of Child Health and Human Development NICHD leads research and training to understand human development, improve reproductive health, enhance the lives of children and adolescents, and optimize abilities for all. For more information, visit https://www.nisht.gov. About the National Institutes of Health, National Institute of Health, National Institute of Health The nation's medical research agency includes 27 institutes and centers and is a component of the U.S. Department of Health and Human Services. National Institute of Health is the primary federal agency conducting and supporting basic, clinical, and translational medical research and is investigating the causes, treatments, and cures for both common and rare diseases. For more information about National Institute of Health and its programs, visit www.nagov. National Institute of Health Turning Discovery into Health Registered.